every step along a metabolic pathway is going to have some sort of enzyme associated with it, helping to speed it up. And it does that by changing energy barriers. It does not affect free energy. Enzymes do not provide energy or remove energy. They only change the structures a little bit. So an enzyme is what's called a catalyst, and a catalyst is something that is going to speed up a reaction without being consumed. So that's really great because it means that we can use the same enzyme over and over and over again. So an enzyme is specifically usually a protein, and enzymes are very specific. So here's an example of a chemical reaction. So we have sucrose, which is a glucose and a fructose, and we are going to do hydrolysis. So we're going to add some water to this chemical bond here with the help of this enzyme sucrase. When you see ASE at the end of a word, that is always a clue that you are dealing with an enzyme. And enzymes are typically named for the substrate that they react with. So sucrase works with sucrose, OSE, meaning sugar. So enzymes are specific. They only work with specific substrates. So the way an enzyme works is it helps the reaction start by helping lower what's called the activation energy. So think about if you're standing on a hill and you've got a boulder and it's not rolling, right? Because it's sitting on the top of the hill and you're right here and you want to roll the boulder down the hill. Once the boulder's rolling, it will be a spontaneous reaction, right? It's not going to take a constant input of energy to roll the boulder down the hill. However, you do have to give it an initial push. So the initial push that you give the boulder is the activation. All you have to do is put some energy into activating it, and then the reaction will go on its own. The boulder will roll down the hill, right? If this is a really big boulder, like this dude is this tall and this boulder is like a 500-pound boulder, pushing the boulder might actually be really hard. So it might require a lot of activation energy. What the enzyme does, let's say we had an enzyme in this little tail, the enzyme is going to make it easier to push it. So maybe this is going to give you some extra muscle strength to help push this boulder. So the enzyme is going to lower the activation energy. So in this example here's delta G, and the reactants are here and the products are here. So that is a negative delta G. So this is a spontaneous reaction, just like pushing a boulder down a hill. We're going to go from AB and CD to AC and BD. However, in the process of doing this, we have to break these bonds and form some new bonds, and there is a kind of transitory state. And so this amount of energy is my activation energy. So you can imagine if I have to go up really high to activate this, this might go really slow. It'll still go because it's still spontaneous, but it'll go really slow. So what an enzyme can do, let's use a different color to show what an enzyme will do. The enzyme does not change the start or the end. What the enzyme can do is it can lower this activation energy so that it's a little easier to get it over that first hump and then get it going faster. So what are you going to be able to do faster? Push a really big boulder or push a really small boulder. So this would be maybe if you had a smaller boulder. So it's going to go much quicker because it doesn't require as much of a push at the beginning. So here you can see that drawn out a little nicer. So this catalysis is the speeding up. It does not affect delta G. Notice that even without the enzyme, and red here with the enzyme, the reactants start at the same spot. And the products end at the same spot on the free energy diagram. So this difference here is going to stay the same. The only thing that's changed is the amount of activation energy that's required with an enzyme versus without an enzyme. So an enzyme is specific. An enzyme will only act on a specific substrate. This area here is called the active site. So this is the region where a substrate is going to bind. And then this is the substrate. The substrate is what binds. So once the substrate binds into the active site, notice that the enzyme has changed shape and it's created a really tight fit. This is called an induced fit, where the chemical groups in the active site physically change the shape of the substrate. And that's how it lowers the activation energy, is it's physically putting stress on the bonds or creating some kind of microenvironment. So this is the enzyme substrate complex because it is literally an enzyme and a substrate. So here's kind of a graphic animation of our A, B, C, D molecule where we're going to physically put them into the enzyme. Notice that this only fits one way. This active site has a very specific shape. You cannot fit these molecules in any other way. So they're only going to fit so that they line up correctly to form the next bonds. So this is the induced fit. We've got our enzyme and our substrates. Sometimes the substrate is one molecule, sometimes it's two, just depends on if the enzyme is breaking stuff or building stuff. In this case, it's doing both. So the fact that this is fit in there has lowered the activation energy, and it's going to be able to change this. So now we have A, C, and B, D, which is the same molecules from the previous reaction. And notice that the way that they fit in there correctly lined up these molecules to break bonds in one spot and form the correct bonds in the products. So after the product has been formed, it gets released. And the great part is that the enzyme is free and available again to do it all again. You can get more substrates and more enzyme and continue to do the same reaction over and over and over again with the same enzyme. They are renewable. So the ways that the active site actually lowers the activation energy, right, this is the activation energy, is by putting things in the correct order. Right, so we saw how it lined up the molecules in the correct orientation so that A and C and B and D are lined up correctly. It physically strains the bonds, so creating that very tight induced fit is going to put a little bit of stress on the reactants so that the products are a little easier to make. Sometimes it creates a microenvironment. This might be temperature or pH or something more favorable to breaking or forming chemical bonds. And sometimes the enzyme actually covalently bonds with the substrate. And anytime you form a chemical bond, you change the structure, and that change in structure can lower the activation energy. So enzymes do not affect the energy of the reaction, the overall free energy. What they do is they speed up reactions. So they affect a rate of the reaction. They make reactions go faster. So if we have an enzyme catalyzed reaction, it can usually be sped up by increasing substrate concentration. So let's draw an example. Here's my enzyme. It looks a little like Pac-Man. So there's 3 enzymes here. If I have one substrate, notice that my substrate is going to fit into this active site. If I only have one substrate for every 3 enzymes, then this reaction will only go at a speed of 1, right? 1 at a time. If I want to increase the rate of the reaction, I can increase the substrate, so increase the number of triangles. So if I increase the number of triangles from 1 to 3, now I can do 3 at a time. So I went from doing 1 at a time to 3 at a time, and that's going to increase the rate of reaction. So let's imagine now my 3 triangles are in the active sites. If I continue to add triangles, is that going to speed this reaction up any more? I hope you said no, because these enzymes are already working. Having 6 triangles doesn't do me any good if I only have 3 enzymes. So when all of the enzymes have their active sites engaged, meaning that I have put a lot of substrate in there to the point where every enzyme is being used, the enzyme is considered saturated. So at saturation, the speed of the reaction will not increase. The only way to increase reaction speed is to add more enzymes. So I want to add enzyme number 4, 5, and 6 to get this reaction to go 6 at a time instead of 3 at a time. The rate of a chemical reaction is also affected by the external environment. So on the scale, you can see that the rate of the reaction is on the side. So the higher it goes, the faster it is. The lower it is, the slower it is. And we can see that a chemical reaction is going to be affected by the temperature. Most of the time, when you increase the temperature, so as we increase the temperature, the rate of the reaction goes up because things are just moving faster. Notice that these reach an optimal temperature. This is a human enzyme. The optimal temperature is about 37 degrees. That's about your body temperature. And this is where your reactions are going to go the fastest. And then watch what happens when we continue to increase the temperature. The reaction rate drops precipitously. Basically, it's a plateau. It falls off really quickly. By the time you reach 50 degrees C, your reactions are not working. This means that you are not alive. Different enzymes work at different optimal temperatures. So for a human, it would make sense that your enzymes would be optimized for your body temperature. This is one of the reasons that we regulate our body temperature so much. A thermophilic, heat-loving bacteria is going to be optimized for a different temperature. So a bacteria that lives at 75 degrees C, which we cannot live at, would be optimized. The enzymes would function here. And those enzymes are not going to function well in our body temperature. Two different enzymes can have two different optimal temperatures. What is happening here when the temperature is dropping off? Well, this is basically the same as cooking your food. You are denaturing the protein. So when the temperature gets too hot for the enzyme, it denatures the proteins. It's going to destroy the secondary, tertiary, and quaternary structures, and it's going to destroy that enzyme. Enzymes also have an optimal pH. So here you can see two examples of human enzymes. So pepsin is the enzyme in your stomach, which your stomach has about a pH of 2. And this is what breaks down proteins in your stomach. And then trypsin is a similar enzyme. It lives in your intestines and it breaks down proteins. And your intestines have a pH about 7.4. The optimal pH for the stomach enzyme conveniently happens to be the same pH as your stomach. And the optimal pH for the intestines happens to be about the same pH as your intestines. So these are going to allow our reactions to go the fastest. So notice as we increase pH, the reaction rate goes up and then it comes back down. Over here, as we increase pH, the reaction rate goes up and then it comes back down. It reaches an optimum. Over here, we get denatured, and then it's going to eventually come all the way back down. So the enzymes in your stomach, when they get into your intestines, do not function. They get denatured and they don't work anymore. Not all of our metabolic pathways are on at the same time. If they were, we would have metabolic chaos. We have enzymes that are working and enzymes that are not working at any one time. And we can do this by either turning on or off the genes that make those enzymes. So if we want enzymes that function, we can just produce them. And then if we don't want those enzymes, we can just turn off the gene and stop making them. Or we can regulate the activity of the enzyme, or we can inhibit an enzyme by just turning it off. A cofactor or a coenzyme are factors that are going to help an enzyme function. So examples of coenzymes are vitamins. Vitamins are required for our metabolism because they help our enzymes function. An inhibitor is going to inhibit an enzyme. So this is going to stop an enzyme from functioning. So here's our enzyme with our little triangle active site. Its normal substrate is this triangle. If I want to inhibit this, what I could do is I could put something else in there, like this ice cream cone, that fits into the active site. So if I have the ice cream cone in the active site, can my triangle get in there? No. So this is considered competitive inhibition. So this is competing for the same active site with the substrate. So this is a competitive inhibitor because it goes into the active site. Non-competitive means that it's not going to bind into the active site. So let's say that this enzyme actually has another binding site over here. Any binding site besides the active site is called an allosteric. Allo just means other, so an allosteric site. So normally, this enzyme would bind with my triangle substrate. An inhibitor comes in and binds here. Let's say it changes the shape of the active site. So here's my non-competitive inhibitor because it's binding to this other site. My active site is still open. Does my triangle fit here anymore? No. Now it's a square shape. My triangle doesn't physically fit here. This enzyme is not going to work. So a competitive is going to bind the active site. Non-competitive is going to bind on some other location that is not the active site, and it makes the active site less efficient, or it makes the active site inefficient. So that would be considered allosteric, is non-competitive, and you can have allosteric activators and inhibitors. So an allosteric inhibitor is going to turn the enzyme off. So here we can see an enzyme complex where we have 4 enzymes here, and they have these allosteric sites between them. So the inhibitor fits into that site really well, and so when the inhibitor is bound here, the active sites are turned off. So this is another form of regulation with an inhibitor. You can also have an activator. An activator can fit in these allosteric sites and then keep it in the on position. So now these active sites are in the proper shape to go ahead and do whatever function this enzyme does. Allosteric, allo just means other, some other place besides the active site. And again, this can be non-competitive inhibition or activation to make sure that the enzyme is on and functioning. One great example of inhibition is feedback inhibition. So this is going to be considered a negative feedback loop. And what happens here is the product of the metabolic pathway turns the pathway off. So this is a really great way to make sure that cells are making enough of the stuff they need, but they're not making too much. As soon as they've made enough product, it turns the pathway off. And then when they no longer have that product, the pathway will turn back on, and then you can make more of what you need. So in this example, the product we are trying to make is isoleucine. Isoleucine is an amino acid that's used as a building block to build proteins. So we need to have isoleucine in our body because we need it to build proteins. We start with a different amino acid called threonine. So again, this is a different amino acid. So our body uses threonine to make isoleucine. So here's our reactant, and here's our product. This is a multi-step pathway. So we have enzyme 1, enzyme 2, enzyme 3, enzyme 4, enzyme 5. And it takes all of these steps to eventually make isoleucine. Isoleucine is my product, and it is a non-competitive inhibitor. So it is going to bind to the allosteric site of enzyme number 1. So when isoleucine binds to enzyme number 1, look at the shape of the active site. It went from being on to now being off. So this is off, this is on. So let's say we're doing this reaction a lot and we make a ton of isoleucine. What that's going to do is it's going to inhibit this enzyme, which is going to turn off this pathway. If enzyme 1 is not working, we're not going to get any of these steps. Like, this is all going to stop, and we're not going to continue to make isoleucine. So the body's going to use up all the isoleucine it has, and eventually when it runs out of isoleucine, it's going to grab this one, pull it out of there, and go use it to build a protein. And once we've done that, it can switch this back in the on position, and now the body can go back to making isoleucine. So we make the product turns off the pathway. We use the product, Once it's all gone, the pathway turns back on. And so it's kind of this cycle of making it, turning off the production, making more when we need it.